Hi everyone. I'm a PhD student at the VU University in Amsterdam. My work is mainly focusing on how to understand the genetic association in biological context. And today I'm going to introduce you a web application I have recently developed, which is functional mapping and annotation of genetic associations. So I'm going to start with a very quick recap what was GWAS again. So we basically start with genotyping a large number of individuals using SNPs array, which nowadays we can tag around 1 million SNPs. And by performing imputation with reference panel, you end up with a maximum of 20 million of SNPs. So in a very simple case, when you have case and control groups in your individual, in your genotyped individuals, you perform the statistical test to see if the occurrence of minor allele in case and the control groups are different from 0. So at the end, you basically get the p-value for every single SNP you have. But as you can imagine, the number of statistical tests we perform is the same as the number of SNPs you have. We, of course, have to correct for the multiple testing. And the golden standard genome-wide significant p-value is 5 times 10 to the minus 8. So whenever you find the SNPs with p-value less than that threshold, those genomic regions are called GWAS risk loci. So the very first GWAS study was published in 2005, and since then, the cost of genotyping dramatically decreased, which allowed us to collect much larger number of individuals. And nowadays, the big consortium for meta-analysis usually use more than 100,000 of individuals. And by increasing the sample size, we also increase the statistical power to detect relatively weak effect size. So, for example, the height study using around 200,000 of individuals, they ended up with identifying more than 100 of risk loci. So we've been performing GWAS in last decade, and nowadays in the GWAS catalog, we have more than 3,000 of studies, including more than 38,000 of unique SNP trait associations for over 600 of phenotypes. So basically, we have a lot of risk loci all over the genome. But however, especially for complex trait, which is highly polygenic, we know that the association of single SNPs is very weak. To detect those effects, we need much larger number of sample size. And luckily, the UK Biobank was just released this month, and the QC2 database contains 500,000 of individuals for more than 1,000 of phenotypes. So the UK Biobank has a potential to identify novel risk loci for lots of human complex traits, and we are expecting more and more GWAS to be published in coming few months. So the question is, what do we get benefit from GWAS results? So ideally, we would like to identify the causal variants from genetic associations that can be used to improve diagnostics or prognostics, or even you might be able to identify novel drug target or biomarkers. However, the association is not the causal. Association doesn't tell anything about causality, and also purely based on the p-value from GWAS, you don't really know anything about the biology. So identifying causal variants from GWAS results is not straightforward. So to overcome this problem, we usually go through the several steps. The first one is to correct for LD, which is non-random occurrence of SNPs. So because of the LD, the most significant SNPs you find from a specific genome risk locus doesn't have to be the one actually causing the phenotype, but there must be the SNPs that is actually causing the phenotype, and that SNPs must have the higher correlation with the most significant SNPs. So we don't want to miss those SNPs just based on the p-value. So the first step is to include all the SNPs that have higher correlation with the significant SNPs. So once you get the list of SNPs you're interested in, the second step is to check the functional consequence on the genes. For example, if you have SNPs on the exonic regions or in the non-coding regions. And there are several softwares that can perform this task. However, more than 90% of GWAS findings are known to fall into non-coding regions. So just knowing that you have GWAS hit in the non-coding region doesn't really help you to understand what is actually going on in biological context. So you still need to annotate biological functions annotations. So there are several data resources that you can use. For example, the CUT score is a deleteriousness score of SNPs, and RegionDB is a categorical score, tells you how likely the SNPs affect the regulatory elements. And there are several EQTL databases. For example, the GTEx have EQTLs in 44 different tissue types. And especially for the non-coding regions, you will also want to check the epigenetic status. The data are available from roadmap and ENCODE. And I didn't bring any database name over here, but the 3D genome, in the field of 3D genome, more and more data is becoming available. So including high-C data might also be another option to map SNPs to the distal genes. So using those functional information at the SNPs level, you can end up with the list of genes you're interested in. And then finally, you need to check the expression pattern in the different tissue types and also the shared biological functions such as pathways. So we've been performing these multiple steps manually, but as you can imagine, that requires you to install different softwares and download different databases, and sometimes you might have to reformat the data each time. So this is very time-consuming and error-prone. So we hope to make a single platform that can perform all of them. So we developed a web application named FUMA that basically automizes 4 steps that I showed in the previous slides into one single platform. So in the FUMA, there are 2 main processes. The first one is SNP to gene, starting from GWAS summary statistics, and we provide list of candidate SNPs with all the annotations and also the list of prioritized genes. And those genes can be passed to the second process, which is gene to func, where we provide you the further biological annotation at the gene level. And another advantage of FUMA is we also provide interactive visualization in the web application, so you don't have to use the external software to just for visualization. So I'm going to go through what FUMA actually does in each process. So in the SNP to gene, starting from the GWAS summary statistics, we first characterize genomic risk loci by correcting for LD. And here we provide you the list of lead SNPs and the genomic risk loci. And all the SNPs which are in LD of lead SNPs are passed to the second step, which is the annotation of SNPs. Here we perform the ANOVA and annotate several variant scores and EQTL and also the high-C. So using those informations, we finally perform the gene mapping. So currently we have 3 different criteria. The first one is positional mapping using annotation from ANOVA, and EQTL mapping, and also the chromatin interaction mapping. So before you perform this gene mapping, you can also filter SNPs based on the annotation you obtain from step 2, and you can also combine different mappings together. You can specify lots of different parameters when you submit the job. And we provide the list of genes mapped by based on the user-defined parameters. So this is just an example how the result page looks like. So we provide Manhattan plot on the top. And the second one is we also perform gene-based tests using Magma software. So this is a Manhattan plot based on gene p-value. And the summary result per genomic risk loci. And all the results are available as a table. And you can also create a regional plot with all the annotation together. And all the results and the plots are downloadable. So this is just one example how you can utilize the EQTL mapping. So this is one of the risk locus on chromosome 14 from schizophrenia GWAS. And from the top, the most top plot is the zoomed in Manhattan plot and the genes, and the CUT score, like RegionDB, chromatin, open chromatin states in the brain, and the EQTLs. So as you can see, the risk locus itself spans multiple genes. So if you don't have any further information, you end up with listing all the genes or you can manually check the function of genes, and you can pick the one that has most interesting function in the phenotype. However, by performing EQTL mapping, we prioritize the single gene which had the EQTL in the brain. So performing different types of EQTL mapping, you can also prioritize genes. And another example is for the chromatin interaction mapping. So the FUMA currently builds in high-C dataset from study of Schmidt et al., which includes 14 different tissue types and cell lines. But I already said that the field is growing very fast. We also provide option to upload the custom chromatin interaction metrics, which is not limited to high-C, but you can also use capture high-C and C5. So the graph right shows the risk loci on chromosome 16 from BMI GWAS. The most outlier is the Manhattan plot. And the second, the blue circle, is the genome coordinates, and the risk loci are highlighted in blue. And inside of the circle, the orange links are high-C link, and the green links are EQTLs. So as you can clearly see, the high-C can map SNPs to distal genes compared to the EQTL. So that might help you to identify novel candidate genes which you have been missed. So finally, once you get the list of genes, you can use gene-to-func process where we provide the gene expression heat map and the tissue specificity by performing overrepresentation tests for differentially expressed genes across different tissue types, and enrichment tests in gene sets, and also the external link to OMIM and drug bank to further investigate the individual genes. So in summary, we optimize the Post-GWAS annotation in a single platform as a web application. So this might be the very first place to stop by to get the very broad overview what's going on in the GWAS risk loci once you get the new GWAS results. But also, if you have a phenotype of interest, there are lots of GWAS summary statistics already available. So you can start, you can perform the FUMA for the available GWAS and start integrating with the research. And for the future updates, we are thinking to extend FUMA to be able to accept the whole exome sequencing study and also the EWAS. And so finally, I would like to thank my supervisor, co-supervisor, and FUMA is available online, so please feel free to visit the website. And I also have a poster this evening at B325, so if you want to know further details, please feel free to visit me. Thank you. You mean like pleiotropy?
It's like same phenotype, but two different causal variants.
In the same region?
In the same region.
Yeah. Well, so it's more like the FUMA is just to annotate what's the functional information available. So it just provides you the options which SNPs you are going to look at further. So it's not removing the information. So you might get multiple SNPs that have functions from one locus. But yeah, we cannot distinguish which is actually causal. But I don't think you are going to miss that information.